some of my thesis work, which is on preparing material which can be used in experiments to evaluate arbuscular mycorrhizal fungi in maize. My thesis has, my thesis has two different chapters with a reverse genetics and a forward genetics approach, but today I'll really just be focusing on one facet of my forward genetics approach. So some background information so that we're all on the same page. Arbuscular mycorrhizal fungi is a beneficial fungal species that lives in the soil, which engages in a symbiotic relationship with plants. This pretty much started when plants moved out of the oceans and began to colonize land, and it's heavily conserved, which indicates its importance to land plant species, such that today about 80% of land plants are capable of this symbiosis. This symbiosis basically entails the fungus helping plants obtain essential nutrients, as well as helping them survive against other environmental stressors in exchange for sugars and lipids and other plant compounds, which the fungus needs to complete its life cycle. One of the most important nutrients that AMF help plants obtain is phosphorus, which is a limited resource for plants here on Earth. And so there's been a lot of interest in trying to see if we could use mycorrhizae to help mitigate the application of phosphorus fertilizer in agronomic systems. So here's a nature review, kind of a general overview of how the symbiosis occurs. So the plant will exude signaling factors, most commonly strigolactones, as a sort of cry for help under nutrient stress. And the fungal spore will perceive these signaling hormones and release their own called MIC factors. And then the spore will induce hyphal elongation towards the strigolactone hormone gradient towards the plant root cell. Upon contact, the spore forms this hypopodium structure and then a pre-penetration apparatus before actually penetrating into the root cells. Upon penetration, the plant cell forms this periarbuscular membrane around the structure called an arbuscule, which is the site of nutrient exchange. So even though the fungal spore is penetrating into the cell, it doesn't actually come into contact with the cytoplasm of the cell. So it's been, so I want to study mycorrhizae in maize because maize is such an important crop. It's grown all over the world. It has it's like $1 billion industry, and we're very reliant on maize. So by understanding how maize utilizes mycorrhizae could help us grow maize better. And because we grow maize in many different environments all over the world, there's the possibility that the relationship dynamics that maize has with different fungal species can be very different depending on the environment that's grown in. So learning more about this and understanding how maize could utilize mycorrhizae could really help in the future with ensuring that we have adequate maize production. And then a really critical point that a lot of people are very interested in with maize is that we could use mycorrhizae to reduce phosphorus fertilizer for maize growth and development. And perhaps most importantly, it's already been demonstrated that there's a connection of mycorrhizae to maize performance, both qualitatively and quantitatively. So in this picture on top, you can see that mycorrhizae susceptible and mycorrhizae resistant plants in the field look pretty different from each other. The plants with no mycorrhizae are yellower and they're not as lush, and they exhibit more leaf senescence than plants that do have mycorrhizae. And you can also see that there's a difference in ear weight between plants with and without mycorrhizae. So this is really exciting because this indicates that there's a real connection of how maize utilizes mycorrhizae in agronomically important traits. It's also been demonstrated that different varieties of maize respond differently to mycorrhizae. So here is a standardized figure from previous research from Rory, which has shown that different modern varieties of maize under sterile conditions where no colonization occurs have differences in their performance, where here he uses root shoot dry weight as an indicator of that. And when we introduce mycorrhizae, we see that some of these varieties undergo a rank change. So all of these varieties, their performance improved when you introduce mycorrhizae to their system, but whether or not the amount of improvement allowed them to perform better than other varieties is what is being shown here. So as you can see, some varieties didn't change quite so much, but what's really intriguing is that this one variety, Ohio 43, shows this like huge rank change where it really responded well to mycorrhizae, such that it became the second best performing under mycorrhizal conditions. So these are all modern varieties of maize. They're all corn. So really the differences in their performance is when mycorrhizae is introduced can be tied back to the small levels of genetic differences between these varieties. And it's really these differences between the varieties that I'd really like to get at and help identify, because by identifying them, we could potentially leverage them in breeding systems in order to make maize more responsive to mycorrhizae. So with that, my thesis, like I mentioned in my title slide, it has this goal of trying to make this material to be used in experiments to measure mycorrhizal response in maize. To do that, I'd like to have mycorrhizae and a no mycorrhizae treatment and elucidate differences in traits that might be different between those two. And so today I'll really be focusing on this B73 Ohio 43 mapping population. But before I jump into that, I really want to emphasize that when I measure traits between a mycorrhizal treatment, I really wanted to be able to measure these traits in field conditions. So there's a lot of incentives for conducting field trials. In the greenhouse, it can be really difficult to grow a large enough sample size to detect some significant signal, whereas in the field, it's pretty easy to grow hundreds, if not thousands of plants. And also, as any grower can tell you, in a greenhouse, the environment is as controlled as possible to give plants an optimal growing environment, but that's almost never the case in the field. Every day in the field, plants experience different weather conditions, different sunlight amounts, and there are temporal and spatial differences in field soil, which presents different stressors to plants in the field. So I really wanted to prepare material that can be used in experiments in real farming conditions that could really derive significant conclusions for farmers. At the same time, with all these incentives for field trials, there's also a really big, it's also been historically very difficult to perform mycorrhizal studies in the field. So native fungal spores that exist in soil make it really hard for you to control mycorrhizae treatments in the field. And while you could attempt to fumigate your fields and sterilize your soil and re-inoculate in an attempt to control mycorrhizae, it's really not feasible to do that because over the timeline of your experiment, the air can carry fungal spores and re-inoculate your field. So this is a really big problem that I needed to address before I could really make any material to use in experiments for field trials. So to address this problem, stood on the shoulders of giants and looked at this paper that was published last year from a former grad student of Rory's, as well as a couple other members of my lab currently. So Maria, whom I'll call Chayo from now on, because that's what we all call her, was able to also make a mapping population that could be grown in field trials, which incorporated treatment with no mycorrhizae. And she was able to obtain this treatment by really utilizing all of this pre-existing knowledge we have about the cellular biological reactions that we needed to know that are required for mycorrhizal colonization. So from that same Nature review a few slides back, this is a broad overview of the very complex cell bioreactions that need to occur in order for mycorrhizal colonization to happen. So she screened through a bunch of these different genes to try and find one that could be viable in inducing a non-mycorrhizal condition. And she was successful in identifying a transposon insertion or multiple transposon insertions in this gene here called castor. And she was able to demonstrate that the second insertion here, castor2, because it's in the first exon of the gene, renders it non-functional. And therefore, you don't see any mycorrhizal colonization. So because this castor protein is essential for mycorrhizal colonization to occur, rendering it non-functional leads to this constitutive phenotype where you don't see any intraradical hyphae or arbuscules present compared to a wild type plant. So by using this castor2 mutation, Chayo was able to make a mapping population, which was successful in determining different QTLs that correlated to dependence or benefit. So here in figures A and B are two different ways that you can think about mycorrhizal response. So some plants are very dependent on mycorrhizae, such that when you remove them, and they're in a no mycorrhizal condition, their performance goes down quite a bit. Conversely, some plants have the capacity to really benefit or respond to mycorrhizae, such that when they are in a mycorrhizal condition or environment, their performance increases greatly. And then there's this idea that there's a trade-off between the two. You can't have the best of both worlds. So here in mycorrhizal conditions, this blue plant performs better than the red. And so this implies that the blue plant is able to better respond to mycorrhizae and increase its performance. But if you suddenly remove mycorrhizae, the performance of this blue plant decreases a lot, whereas the red plant doesn't. So this implies that the red plant was just not very dependent upon mycorrhizae in the first place. So Chayo was able to perform QTL mapping and identify genetic markers that could actually be distinguishable between dependence and benefit. So here in figure C, she identified this QTL where when you have mycorrhizae, there's really no big difference in effect size, but when you remove mycorrhizae, you suddenly see that there's a difference in effect size between the QTLs, depending on whether you're derived from the CML or the W22 background. And because you don't see this difference in effect size until you move to no mycorrhizae, this implies that this QTL is correlated with dependence upon AMF. And then you get the converse story on this side. So when you don't have mycorrhizae in the resistant treatment, your QTLs aren't significantly different from each other. But when you introduce mycorrhizae, you suddenly see a difference in QTL effect size. And because you only see this occurring when you move to mycorrhizae susceptible or plus mycorrhizae, this implies that this QTL is correlated to some benefit or responsiveness. So a quick summary of Chayo's paper. A lot of the ideas that I'm pulling from her paper to use in my thesis work is that she's able to have these two treatments for her mycorrhizal study of susceptible and resistant. And the way that she obtained her resistant treatment enabled field conditions or field trials, which is also something that I wanted to do. A really important note is that she grew these mutants in both greenhouse and field conditions under high input conditions, and showed that they're not impaired in normal plant development. So it's not, so she concluded from this that there aren't just too many negative pleiotropic effects from this castor mutant that just puts the mutants at a disadvantage compared to their wild type counterparts. When they're grown under sufficient nutrient conditions, the plants are pretty much indistinguishable from each other. And so it's really only when plants are put under stress that their dynamic with mycorrhizae comes into play. And that's really what we're trying to hone in on. Like, how do maize plants under stress utilize mycorrhizae in order to ameliorate that stress? And probably the most exciting part, I think, about her paper is that she's really able to identify different markers tied to dependence or benefit. I think it's really cool that the way she went about enables not only her to identify QTLs that are important to mycorrhizae, but also the context of how they are important to whether they stand to benefit or whether they stand to depend on mycorrhizae. So with that, the goal of my thesis was to generate a mapping population to identify these markers that are tied to responsiveness. So if you remember from a few slides back, Ohio 43 was one of those varieties that responded very, very well to mycorrhizae. So I wanted to elucidate the genetic markers specifically in the Ohio 43 variety to try and identify what those markers were that enabled Ohio 43 to perform so well compared to other varieties like B73, which didn't really improve that much with mycorrhizae. So here I'm incorporating both castor and another gene, Pollux, which Chayo also demonstrated was essential for mycorrhizal colonization to occur. I incorporate both genes instead of just one in order to account for pleiotropic effects that castor might have. So last winter in the greenhouse, I crossed homozygous mutants for castor and Pollux, which are in the W22 background, to both B73 and Ohio 43 plants. And then last summer, we planted that F1 seed in the field and selfed it down to create a segregating stock for each of these 4 mini populations. So this summer, we'll be planting this F2 seed and we'll be identifying wild type B73 and Ohio 43 plants, which will be mycorrhizae susceptible, as well as homozygous mutants for castor and pollux, which will be mycorrhizae resistant. So by identifying these individuals, we'll self them down one more time and create F23 families, which can then be used in field trials to evaluate mycorrhizal performance. So I'm hoping to hit 800 F23 families, which means I really want to be able to identify about 1,600 homozygous wild type and mutant plants in order to ensure that I get enough years back. So a really important feature of this mapping population for its success, that its success is dependent upon, is that I accurately identify the wild type and homozygous mutants in this summer's F2 generation. So the way that I've been doing that before, the way that I've been tracking the genotype so far has been by traditional genotyping by PCR. So if you remember, Chio had identified a transposon insertion in castor and pollux. So I could perform the traditional genotyping by PCR using a forward and a reverse primer along with a primer to help identify whether or not the presence of the transposon is there or not. So here I'm showing you some genotyping results. So I run my 2 reactions with my forward and reverse, and then also one with my mutant primer in order to determine what the genotypes of my individuals are. So here, individual A from family 1 will be a heterozygote. Individual B will be only a wild type plant. C is also a heterozygote, and D will be a homozygous mutant. So this is one way that I can track, that I can accurately identify the wild type and homozygous mutants this summer. But another way that I could do it is also, instead of trying to identify whether or not my transposon is present, I can just try and identify whether or not the castor or pollux gene is derived from the W22 background, or whether it's from the B73 or Ohio 43. So the idea here is that anytime that the mutant castor gene is present, it can only be from the W22 background. If it's the wild type copy, then it must be from the Ohio 43 background. And the same is true for B73 as well as for pollux. So the rationale here is that if there were SNP differences between my 3 varieties that are either within or nearby the castor and pollux genes, I don't need to test whether the transposon is present. I just need to know that the copy is derived from the W22 background in order to know that I have a mutant copy of castor and pollux. And one of the really great things about working in maize is that there's a lot of genetic resources out there, one of which is a tool called SNPversity, which lets me look for exactly this. So I can go to the castor and pollux gene and look between my 3 varieties and look for specific positions where my W22 base pair will differ between Ohio 43 and B73. So I don't remember my next slide. Okay, yes. So because I have identified SNP differences between W22 as with B73 and Ohio 43, I can use the specialized type of PCR called CASP genotyping, which is an acronym for competitive allele specific PCR. The way this works is that you have two forward primers that differ only in a single base pair. The two primers have different fluorescent tags on them, and you can run your PCR, which will carry either G or T, and such that when you run your PCR, only one of your primers should bind, whichever one has the higher affinity based on which one matches at that single base pair location. When this PCR reaction occurs, your DNA polymerase will cleave the fluorescent marker and emit a signal, which is detected and interpreted by a computer. So you can feed samples in and get a very easy output. And there's a lot of benefits to using Cas genotyping, which is why I really wanted to make this a viable method of genotyping for the summer instead of the traditional genotyping by PCR. So based off of my SNPversity searches, I identified 10 different candidate SNPs that could be used to distinguish Castor and Pollux genotypes in my F2 seed. Like I mentioned before, I wanted to identify about 1,600 of them. So it's really important that my genotyping be accurate and relatively fast. And I was able to see that, yes, my Cas genotyping with the candidate SNPs I selected worked. So here I'm showing you one of the SNPs that I chose that is polymorphic at this position, which can either be G or A. The W22 sequence, the W22 version of Castor should call it at A, whereas B73 and Ohio 43 should call it at G. So when I run my samples, I do see that my controls behave the way I expect them to, that W22 does call it A, whereas B73 and Ohio 43 call it G. So then I can start feeding in samples, which I know will either be the W22 B73 mix or the W22 Ohio 43 mix, to be able to identify whether or not these individuals are wild type, heterozygotes, or homozygous mutants. Another really nice feature of Cas genotyping is that it's very easy to visualize your data. So here, this is for the same SNP in Castor. You can see that the homozygous mutants, which call it AA, cluster together, whereas wild type plants cluster on the other side, and heterozygotes, which have one copy of both, cluster somewhere in the middle. So this is really exciting that the Cas genotyping works and accurately identifies the genotypes of my samples. At the same time that I was evaluating whether or not Cas genotyping would work, I also wanted to know if my gene was segregating as expected. So this F2 seed is a heterozygote that's been self, so I'm expecting a Mendelian ratio of 1 to 2 to 1 for my wild type heterozygotes and homozygous mutants. My chi-square values for both of my genes, Castor and Pollux, are less than my critical values, so I can confidently say that yes, they are segregating the way I want to, the way that I would expect them to. So I feel very confident moving forward with this F2 seed for this summer's planting, where I'll be selfing down about 1,600 plants. Something that I wanted to really emphasize here is that Cas genotyping works better than traditional genotyping by PCR. So the same individuals that I optimized my Cas genotyping for, I also conducted traditional genotyping on them. And I think you could see pretty much right away that the traditional genotyping is a lot messier to interpret than the Cas genotyping was. So again, the traditional genotyping requires two reactions where I'm trying to detect the presence of the transposon. And one of the caveats of traditional genotyping is that one of the caveats is that I have to interpret no band without knowing whether I should not be seeing a band or whether my reaction has just failed. So here for this very first sample, even though it's very ugly to look at, I would interpret this as no band here and a band here. So based off of that, I would interpret this individual as a homozygous mutant. However, I don't have any indicator whatsoever whether there really isn't supposed to be a band here or if this reaction just happened to fail, and that if I reran it, I would see a band. And if I did see a band, that would change my genotype interpretation. So because I ran these same samples with cast genotyping as well as this traditional genotyping, I was able to compare the accuracy rate between the two. And I saw that between my samples, there's a lot of variation in how accurate this traditional genotyping is, which is sometimes as low as almost a quarter of the time, which is just abysmal. And this is really not what I was looking for this summer when I needed to score hundreds of plants. So I'm really glad that I was able to get the cast genotyping to work because it's a lot more accurate than the traditional genotyping. In comparison, if your reaction fails in cast genotyping, you won't actually get a signal because your results are dependent upon that fluorescent marker being cleaved. So if your reaction just fails, you actually will see that there's very low signal for your sample. And sometimes your samples aren't perfect, and you'll see that the algorithm won't make a call for you, but it's a lot easier to look at your sample batch as a whole and see where this unknown call fits relative to the rest of your samples, which I think is really nice. Okay, so I've talked a lot about how I'm making this mapping population and how I'm optimizing the genotyping, but I just wanted to spend a little bit of time talking about at the end how exactly this mapping population will be used to measure mycorrhizal responsiveness. So this summer, after selfing down, we'll have completed the 800 or so families, which can then be planted in field trials and evaluated for whatever traits we're interested in. Once we have this trait data, we can see if any of it correlates to mycorrhizal treatment, so susceptible or resistant, as well as different to genetic differences between the two varieties that I use, B73 or Ohio 43. However, what I'm really interested in is the interaction between these two loci. So this interaction variable is really asking the question of whether or not mycorrhizae influence this trait differently between my two varieties of B73 and Ohio 43. And it's really by identifying these interactions QTLs that will be indicative of QTLs that are tied to maize responsiveness to AMF. Okay, so in summary, I spent a good chunk of my master's time here making this mapping population, which can be used in field trials to measure mycorrhizal response. I optimized cast genotyping to make life a little easier this summer. And I'm very excited to be moving forward and planting this F2 seed. I think I've been telling my lab mates for quite a while now that I can't wait for planting to happen because I can't wait to plant these F2 families. And my last point is just that I really only had time today to talk about my B73 Ohio 43 population, but I did have another population that I got to try my hand at where I'm asking a similar but distinctive question about whether or not maize responsiveness to mycorrhizae has changed over time. And then as well as a totally different approach in where I was looking at genes that I thought would be important in mycorrhizal response and generating some mutants in those to begin evaluating those mutants for phenotype. So if you have any questions or are curious about the other stuff I worked on, I'd be more than happy to answer some questions about that too. But with that, I'd just like to say thank you to everyone who's helped me in my time here. So my committee, which gave me a lot of great insights to develop my thesis, as well as my growing lab, who's been so helpful in explaining science to me, as well as just doing grunt work with me, both in the lab and in the greenhouse and in the field, as well as other people who've helped me in various many ways. And with that, I'd be happy to take any questions.
Anybody have any questions for Liza? Feel free to either use the chat or unmute yourself.
The quick question, how much does the CAS cost versus normal PCR?
Yeah, so you can do CAS genotyping in-house as well. You just need like a real-time PCR machine to do it. We just, since Rory and I were new here, we haven't set that up yet, which is why we're licensing out to a third party right now. I think it's not significantly more expensive. I think based on the sample size we're going to, it's like 20 cents more expensive to run our samples through this third party company with CAS genotyping. But I'm sure if we were to set it up and be fully established, it'd be cheaper for us to do it in-house.
Lisa, I have a question. This is Surinder.
Yeah.
Hey, nice talk. Thank you. I wanted to ask you that when you were making this, or you had this mutant in the W22 background, and then you have non-W22 background where you don't have the mutant. So I mean, the actual W22 background. Are there any clean differences in the phenotypes?
Of W22 plants that carry the mutation?
With or without the mutant gene?
Yeah, so I don't have a figure pulled up, but that's what Chao's paper was trying to factor in. So she identified those transposon mutants in the W22 background because these are mutator from uniform mu. And so she grew the homozygous mutants in W22 versus wild type W22 plants. And she was able to demonstrate under like high nutrient conditions that there's no difference in performance between them. So it's really only when she introduced stress, like, you know, like low phosphorus or water stress or something, that she was able to start seeing a performance difference, which would indicate that there's some use of maize utilizing mycorrhizae to ameliorate stress. And so these mutants aren't able to utilize mycorrhizae, and so they now perform worse.
Yeah, so I guess in that case, you won't be able to use any phenotypes, you know, between, you know, let's say if you see any differences in the W22 carrying the mutation and Ohio 43, which is the wild type gene. So you won't be able to see any phenotypes in order to link your CASP marker a little bit more cleanly and say, you know, I think this plant that does have the Ohio 43 gene, it is Ohio 43.
Yeah, so that's exactly what we're, so that's kind of why we've incorporated W22 with B73. So the idea here is we're going to compare Ohio 43 to W22 and then B73 to W22 because they're tied with a baseline of W22 that allows us to compare B73 wild type with Ohio 43 wild type, which Rory's prior research has shown, you know, Ohio 43 stands to have greater response to mycorrhizae than B73 does. That's why we can just mix, yeah. That's why we're trying to incorporate like two different parents, but we're a little limited because we have the castor and pollux mutations reside in the W22 background, so we had to include it.
Great, good luck. That's good. See you later in the fields.
Yeah, I'm looking forward to it. Thank you.
Hi, Lisa. Thank you for your presentation. It was very interesting.
Thank you.
I wanted to ask you, so I think there's a lot of interest in this whole area of mycorrhizae and how we can benefit better from them. But from your presentation, I understand that even within one, so you said 80% of plants, land plants have mycorrhizal associations, but within a species, there is still, are these plants just different in mycorrhizae in that they are not, are they not colonized, or the ones that don't rely much on the mycorrhizae, or are there are they colonized but they don't benefit from them, or how does that work?
Yeah, so that's a really big question that I think a lot of microbiologists really want to answer. So there's so much variation in the kind of relationship that a land plant might have with mycorrhizae. So on the figure that I was showing earlier, there's this idea of like some plants, even within a species, might be more dependent on mycorrhizae, right? That they rely on mycorrhizae for their development, whereas some plants don't really need mycorrhizae to grow and develop, but if you give them mycorrhizae, they respond really well and can grow bigger and better. And then there's also variation on the fungal side, right? So this isn't just one single species of mycorrhizal fungi. There are like hundreds of them, and they're present at different densities in these microbial communities in the soil. And then different environments will have different species. So it's a really big web to try and untangle, I think. And this is really, this mapping population, in order to be as detailed as possible, I think is really just scratching the surface of how, from the plant side, plants might have genetic markers which enable them to respond better to mycorrhizae.
Thank you.
Yeah, of course. Yeah, Mung knows a lot more about microbial communities of AMF if you're interested on that side.
So perhaps I can ask another question.
Yeah, of course.
Since some varieties or cultivars are not really dependent on the mycorrhizae, you say, would this affect their performance in, say, since mycorrhizae are known to be especially, can be important for phosphorus uptake, would this then affect their ability of these cultivars, one cultivar to do better in a low phosphorus environment than another one?
Yeah, one of the reasons why, one of the really exciting treatments I want to have this mapping population in under the future is we have high phosphorus and low phosphorus plots at the farm here, and I would really like to see if there's something going on in performance when you have those kinds of conditions. I think in general, it might be a little strong to say that because different plants might adopt different strategies for nutrient acquisition, right? So, you know, plants are not obligated to form the symbiosis. So you can grow maize plants, you know, to full maturity and development and get a good yielding ear even in sterile soil, as long as you're feeding it enough nutrients. So whether a plant decides to adopt the strategy of like more root growth in order to acquire the required nutrients, or whether it decides to engage in higher levels of the symbiosis in order to get the same effect of acquiring its nutrients, is something that we're still not sure how it's regulated by plants. But it was really exciting to see some of Rory's prior research that showed that there is this variation, that some plants do utilize mycorrhizae more than others.
Great.
Any other questions?
Hey Liza, it's Lily. So I was wondering about the SNPversity. Could you explain a little more about that, please?
Yeah, so there's this amazing website called MaizeGDB, and it's basically a powerhouse for everything corn related. One of the tools on the website is called SNPversity. It's like SNP plus diversity put together. So what's really awesome about maize is that there are many different genomes of different varieties that have been sequenced down to the base pair level. So this tool, SNPversity, just lets you pull different maize varieties and compare at the base pair level the differences between them. So it's a really clean interface and easy way for you to identify polymorphic SNPs between different varieties, which is exactly what I used it for cast genotyping.
Okay, great. Thank you. Lovely talk, by the way.
Thank you. Okay, so if there are no more questions, I'm gonna stop sharing my screen. Thank you so much, everyone.
Thank you, Lisa.